All right, welcome to this explainer. Right now, literally all around the globe, there's this invisible, high-stakes war going on. And we're not talking about tanks or drones here. No, this battle is fought with steam, toxic gases, and actual radiation. It's happening every single day inside pharmaceutical manufacturing facilities. The ultimate goal? To keep the medical products we all rely on completely, 100% safe for human use. So today, we're going to dive deep into the intense, exact science of pharmaceutical sterilization.
So think about this. Is a clean-looking medical instrument actually safe to use on a patient? Well, our eyes can easily deceive us. You look at a shiny stainless steel scalpel or a totally sealed glass vial of eye drops, and they might look perfectly pristine. But zoom into the microscopic level, and they could be absolutely teeming with bacteria, viruses, and highly resistant fungal spores. In the medical world, simply looking clean just doesn't cut it.
Which brings us to our core definition, right? Sterilization. This is the complete elimination of all viable microorganisms. We're talking about the pursuit of absolute zero. Unlike your basic everyday cleaning, true sterilization has to achieve a literal 100% kill rate. And crucially, it's got to be sporicidal. Spores are basically these armored survival pods that bacteria form when they're under stress, making them insanely difficult to destroy. If a process doesn't kill the spores, well then it's not sterilization. Period.
So, to understand how the industry achieves this absolute zero, here's our roadmap for the explainer. First, we'll look at sterilization versus disinfection. Second, terminal versus aseptic processing. Third, the actual arsenal, heat and gas. Fourth, we'll cover radiation, specifically beta and gamma. And finally, validation, how we actually prove it works.
Okay, let's jump right into Section 1, sterilization versus disinfection, and really understand the limits here.
The absolute most crucial point here is that these two terms are not interchangeable. People mix them up all the time. Disinfection, that's really just about reducing microbes. If you disinfect a surface, you've lowered the microscopic population down to a level where, handling it won't cause a major infection. But the count is never actually zero. Those super resilient spores, they survive. Sterilization, on the flip side, is completely sporicidal. It leaves the item 100% free of all living microorganisms. And look, when a product is about to be injected directly into your spine or dropped into your eye, only absolute sterilization is going to do the job.
Moving right along to Section 2, terminal versus aseptic processing. Let's break down the manufacturing strategies.
So right now, there is this massive growing demand out there for small molecule parenteral products. We're talking about everyday lifesavers here. Heart medications, saline bags, eye drops. Obviously, these products have to be completely sterile, and the safest, most foolproof way to get there is through a method called terminal sterilization. This essentially means the liquid formulation is mixed, it's filled into its final tightly sealed container, and then that entire finished package is sterilized as one single unit.
Now, the math behind this level of safety is honestly fascinating. We measure it using something called a sterility assurance level, or SAL. Because terminal sterilization treats the final sealed product, it achieves an SAL of 10 to the negative 6. To put that into perspective, that means there is only a 1 in a million chance that even a single viable microbe survives. It's no wonder regulatory agencies like the FDA strongly prefer this method. But here's the catch. Some drugs, especially complex biologics, would be completely destroyed by the harsh, extreme conditions of terminal sterilization. For those sensitive products, we have to use aseptic processing, which does carry a slightly higher risk level of 10 to the negative 3.
And because we can't just blast the final sealed product to sterilize it, aseptic processing is kind of like a massive, highly choreographed puzzle. It requires every single individual component, the actual drug, the glass vial, the rubber stopper, to be sterilized totally separately, and then carefully assembled inside a highly controlled clean room. Liquid drugs are physically pushed through microscopic, sterile filters just to catch and remove bacteria. And the human operators, they have to be covered head to toe in sterile gowns, hoods, and goggles. Sometimes they even have to work through restrictive glove ports built into the glass, simply because, well, we humans are the single biggest source of contamination in the room.
All right, on to Section 3, the arsenal, heat, and gas. Let's check out the actual weapons used in this fight.
The absolute most trusted workhorse in the entire pharma industry is the steam sterilizer, better known as the autoclave. But listen, it is not as simple as just boiling some water. It's a precisely engineered 4-step process. Step 1 is make or break, air removal.
Air actually acts as an insulator, so it's the mortal enemy of sterilization. They use these powerful vacuums to suck it all out. Step 2 introduces saturated steam under intense pressure. Step 3 is the sterilization hold, which usually means baking the load at 121 degrees Celsius for a good 15 to 30 minutes. And finally, Step 4 exhausts all that steam and pulls yet another vacuum to dry the load completely.
Now, if you're a science nerd like me, the physics behind why this works so incredibly well is just awesome. It's actually not just the high temperature of the steam that kills the bacteria, it's the phase change. When that super pressurized steam hits the cooler surface of the medical load, it instantly condenses back into liquid water. And that exact moment of phase change releases a massive burst of energy known as the latent heat of condensation. This energy rapidly transfers directly into the microbes, violently denaturing their proteins and completely disrupting their cellular enzymes. It is incredibly fast, and it is brutally uniform.
But, as you can probably guess, not everything can survive being blasted with superheated high-pressure steam. For those heat-sensitive medical devices, the industry historically had to rely on ethylene oxide, or EO gas. But EO comes with a massive drawback. It's highly toxic. We're talking real cancer risks for workers and surrounding communities. Plus, it requires these painfully long aeration times just to make sure all that toxic gas is fully removed from the product. Fortunately, today we're seeing a huge paradigm shift toward hydrogen peroxide gas plasma. It's highly effective, but so much safer, because once the sterilization cycle is done, the gas literally just breaks down into harmless water vapor and oxygen. Talk about a game changer.
Which brings us to Section 4, radiation. Beta and gamma rays. This is how we tackle sterilizing items that are already totally sealed.
I gotta say, radiation is particularly incredible because it can sterilize a product after it has been completely sealed inside its final packaging. By directing ionizing radiation right at the product, that energy just passes straight through the cardboard and the plastic like it's not even there. And when it hits any bacteria or fungi trying to hide inside, it literally shatters their DNA. Without intact DNA, those microbes are instantly inactivated. They can never reproduce again, which leaves you with a completely sterile product that is ready to ship straight to the hospital shelf.
There are two main types of radiation used for this, beta and gamma, and they operate very differently from one another. Beta rays, or E-beams, are actually generated using electric current and a particle accelerator. They deliver a massively high dose of radiation in mere seconds, which is awesome for the materials, but they have a pretty low penetration depth, so they really only work on individual boxes. Gamma rays, on the other hand, are emitted from a decaying radioactive isotope called cobalt-60. They have a much lower dose rate, meaning the product has to sit inside this heavy irradiation chamber for hours. But man, their penetration power is absolutely immense. You can sterilize entire, massive pallets of product all at once with gamma rays.
All right, all of this high-tech science brings us to our final stop, Section 5, validation. Proving it works and assuring we actually hit absolute zero. You see, to actually guarantee your safety, scientists first have to evaluate something called the bioburden. That's just the natural starting number of microorganisms already chilling on the product. By plotting this initial microscopic head count against the known radiation resistance of those specific microbes, they can draw out this precise graph you're seeing. It tells them exactly how high the radiation dose or the heat cycle has to be to push the survival probability all the way down to that crazy 1 in a million standard. They are literally mapping out the mathematical death of the bacteria.
And to ensure the equipment actually hits those mathematical targets every single time without fail, engineers follow this incredibly rigorous layered testing protocol. First up is design qualification, basically ensuring the blueprint meets all the safety standards. Next is installation qualification, proving the machine on the floor was built exactly to those blueprints. Then comes operational qualification, where they take every single sensor, valve, and alarm and test them at their absolute extreme limits. And finally, the ultimate test, performance qualification. This involves running actual test loads that are deliberately spiked with tens of thousands of highly resistant bacterial spores, just to prove, beyond a shadow of a doubt, that the machine can completely obliterate them.
Which leads us perfectly to this foundational principle of the entire industry. In pharmaceutical manufacturing, assumptions don't count, only proof does. Every single time you or a loved one receives an injection, uses an eye drop, or undergoes a surgery, you are directly relying on an incredibly complex, highly validated chain of science that is working overtime to protect you from an invisible threat. Today we've seen how heat, toxic gas, and literal radiation are precisely controlled to achieve that absolute zero. So I'll leave you with this to ponder. In a microscopic world that is constantly mutating and fighting to survive, is a medical instrument ever truly safe until we can mathematically prove its absolute purity? Thanks so much for joining me on this explainer, and keep questioning the unseen world around you.